channel Anjan Bios. We are going to discuss about the structure of ovule, the Megasporengium. You see, this is the Megasporengium. We know gynoecium represents the female component of a flower. The pre-unit of the gynoecium is called pistil or carpel. As you know, a pistil consists of three parts: stigma, style, and ovary. You see, this is the structure of a typical pistil. This portion is called stigma. A long style is here. This is this one style, and this is the ovary. This one, and inside the ovary, this swollen part is called ovary, and this ovary has ovarian cavity with one or more chambers. You see, there are several chambers are here. These chambers are known as locules, and it is made up of ovule-bearing parenchymatous cushion called placentae. In single, it is known as placenta. An ovary, this swollen portion, may contain sometimes one ovule as in mango or several ovules as in watermelon. Actually, the ovule, which is known as fusar seed, it is an integumented megasporengium. This structure, whole structure, is called megasporengium or the ovule. The body of the ovule consists of a mass of parenchymatous cells called nucellus. This structure, this is the nucellus. This nucellus is surrounded by two multicellular integuments. You see, this is one and this is two. Interestingly, these integuments leave a narrow pore or passage at one end. The end where this narrow passage or integument is not fully covered is called micropylar end. This one is called micropylar end, and opposite to micropylar end, this end is called chalaza. So if micropylar end here, then chalaza end will be here. That is, the pore or space where integuments not covered and leave this portion is called micropyle. The ovule is attached to placenta by a stalk. You see here, this one, this small stalk is here. This stalk, this one is called funicle. This stalk, this portion is called funicle. And the body of the ovule, fused with the funicle, a short appendage, this is called hilum. This portion is called hilum. And a longitudinal ridge, this black color ridge, is present in the outer integument through, and this supply nutrition to the ovule, and just it behaves just like conducting tissue. This is called raphe, this one. And the whole structure, this integument, the nucellus, bear this embryo sac. This one, this is the embryo sac. This one, generally an embryo sac contains 8 nuclei but 7 cells. The 3 nuclei present towards the micropylar end, 3 nuclei present towards the salazal end. The nuclei present towards the micropylar end is called egg apparatus, where one egg and two supportive cell or nuclei, this is called synergids, and opposite to micropyle, that is in salazar end, 3 nuclei are present. These are known as antipodals, and 2 central nuclei present within the center of the embryo sac, and this 2 nuclei is called polar nuclei. So in a mature embryo sac, we have 8 nuclei, 3 towards the micropylar end, 3 towards the salazar end, and 2 in the center, and all finally form cell. The cell will form and finally behaves as cell. So a 7-celled, where the middle one is diploid and salazar end and micropylar end cells are haploid, 7-cell, but it contains 8 nuclei. This is a mature embryo sac. This embryo sac, it is a structure before fertilization. Then in the next class, in the next video, we are going to discuss how this megaspore or this embryo sac will form inside the ovule. This is called megasporogenesis. Thank you.